This is the Convergent Science Network podcast. Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
This is Paul Verschure for the Convergent Science Network podcast here at the Barcelona Cognition, Brain and Technology Summer School of 2018. And I have with me Elena Galea. Welcome to the podcast, Elena.
Thank you very much. Happy to be here.
Great. And your talk was about astrocytes, and also was immediately instructed not to call them glia anymore because they're astrocytes. So maybe this is also a good starting point for the discussion, right? So why do we have to drop this notion glia and focus on astrocytes?
Well, because it doesn't make any sense to classify cells between neurons and non-neuronal cells, because the non-neuronal cells are so different among themselves. This is like saying in the whole body that the body is divided into heart and the rest of the organs, intestines and liver. It makes no sense because those organs are so different. They have different problems and different features. So you have to focus on them individually. So the non-neuronal cells include cells that are so different in their functions and in their molecular phenotype, in their contribution to pathology, contribution to higher brain functions, that just classifying them, just putting them together in a single concept, it just doesn't force us to look into the details. I think details are very, very important. Names are very, very important because they guide research and they guide the understanding of problems. So it makes sense 1 century ago to say neurons and the rest of the brain. Now it doesn't make sense any longer.
Okay, so basically you're saying we have many cell types that build the brain. Some of those are spiking. They generate action potentials. Those are the ones we call neurons. But then the many other cells, which is a very variable set of cells that formerly were known as glia, the non-neuronal cells, but it is not in any way descriptive of this variability. And then among this highly variable set of cells, we have a subtype called astrocytes, right? So what then sets the astrocytes apart from these other non-neuronal cells?
Well, first, molecularly, they're different already. They have a different identity. And they carry functions that the others don't. They don't generate myelin, like oligodendrocytes. They don't deliver blood, like the vascular cells. They don't protect neurons from some danger like microglia. So at least they don't do what other things do. And perhaps they do things like providing lactate to neurons, removing potassium from the extracellular medium. They transform glutamate into glutamine. So they have very, very specific functions that they only astrocytes do and none of the rest of the cells do. So this is what is important to focus on them.
But they stand apart also on purely morphological grounds?
Morphologically, yes, depending on what comparison you carry out. Because there are other cells in the brain called NG2 oligodendrocytes. They are like between astrocytes and microglia. So if you compare an astrocyte with a neuron, they're very different. So neurons have their dendritic spines and axons, while astrocytes are very, very complicated cells, bushy cells with very intricate distal processes. But NG2 have also primary, second, tertiary processes and some dense distal processes. So they are intermediate. And in microglia, they have very long processes and they don't have the distal processes. So it seems that there is a morphological gradient in the brain that differentiates neurons from microglia, but not so much NG2 from astrocytes. Vascular cells are very different from the rest of the cells. Oligos are very similar to neurons in many ways. So depending on the comparisons you establish, that the morphological differences are larger or smaller. But yeah, morphologically, they're very, very different.
Okay.
Absolutely. And molecularly and morphologically, very different.
So in the human brain, we have about 90 billion neurons, right? According to the most recent counts. Do we have then 90 billion non-neuronal cells?
There is what is her name? Herculano.
Yeah.
So those calculations, that's a fantastic word. That there are papers. She has analyzed that with very objective technology, and so the answers are there. I don't think that. I think the answer is that equal amount of neurons and not
50-50.
I think it's 50-50. Depends on the animal. Depends on the area of the brain. So I don't think it's 50-50. So she has calculated those numbers.
But now, of the non-neuronal
do you think this is relevant? Like numbers of
just plenty of ratios. Like
ratios.
Let's say, if it's like, well, you gave some numbers actually in your talk, right? Where you said that, where do I have
on average,
in humans,
on average, like 2 astrocytes per each 10 neurons. I think that's an average calculation. On average.
In mouse, it was 1 to 20 neurons per astrocyte, right? You said.
Or 1 to 20 or 2 to, is in that.
Isn't that range for humans as well?
Yeah, probably.
So that would be, let's say.
But what I meant is that there was for many years the notion that astrocytes were 10 times more abundant than neurons. Exactly. And first, this is not true. And I don't think the numbers are so relevant in the sense that the fact that you have more of something makes that something more relevant, perhaps, and perhaps not necessarily. I think it's the function that is more relevant. You may have something that is just present in very low amounts, but it's for some reason incredibly important for the function of the brain.
Okay.
So numbers are interesting, but I don't think that is so relevant. And definitely, astrocytes are not 10 times more abundant in the brain than.
But of the non-neuronal cells, how abundant are the astrocytes compared to the other non-neuronal cells?
I don't have the numbers. Okay. Herculano has them.
But just in your imagination, astrocytes are a relatively small subpopulation of cells making up the brain, so they're not that dominant. Right? So, and then if you look at the non-neuronal cell types, is there anything special about their distribution in the nervous system if we go for more primitive parts of the brain, like the brainstem and the spinal cord, to more frontal areas? Do you see any kind of differentiation across these non-neuronal cell types?
I don't think that this has been analyzed. We spoke about tiling, and I think tiling it's a feature of astrocytes that neurons don't have. And perhaps there is more tiling the more frontal we go. So the more developed the area, the more tiling.
More regular, if you want.
More regularly and perhaps tighter. So the tiling function, whatever it is more developed. That's perhaps the case. But those kind of analysis haven't been performed.
And as from an evolutionary perspective, if we go to, say, primitive vertebrates, let's say lamprey or fish, or we go to invertebrates, do we see any kind of patterning of the non-neuronal cell types?
I'm not sure about that. I know the difference between mice and humans, that astrocytes are more complicated structurally in humans than in astrocytes, and there are more subtypes. And I don't know exactly, but I think in invertebrates, there are astrocytes, but I'm not sure about their localization or whether they are.
But it also means people might not have really looked at that.
No.
Okay. So that's interesting, right? Because one thing that you also made clear in your talk is that the whole idea already of the morphology of astrocytes has changed a lot, right? Because initially, because of the immunocytochemistry available.
Because there are better techniques now to see astrocytes,
Initially, they looked like very sort of punctuated star-like
cells, it's mostly with long processes. Yeah. And they're not. They're like balls of a lot of hairs. That's the way they are.
Right.
And that's probably indicative of their function and the way absolutely.
So, and that's also some of your own work and that you showed. So, to get to the tiling, so it looks like we have these bushy astrocytes that are forming sort of a tessellated pattern throughout the brain, which is structured in a three-dimensional way. So do you also think this tiling is highly structured in 3 dimensions?
Oh, absolutely.
Like bricks, like we should hit the boxes?
Absolutely. It's like a Voronoi tessellation. Absolutely. It is. I know. We have published a paper actually where the tiling of astrocytes it's really well modeled when you do Voronoi tessellation of the brain. So you take the brain and you just do this kind of tessellation from... You just have seeds there, and then you establish the distances between the seeds, and you draw walls there, and that way you divide the brain like a given volume in different Voronoi volumes, and it's identical to the tessellation, to the natural tessellation of astrocytes, indicating that I think this is indicative of the way they grow. So probably when they grow, either from radial glia or from clonal expansion from local astrocytes, they sort of grow until they find another astrocyte. And that's actually how Voronoi tessellation occur and develop.
But that's for the neocortex, mainly, no?
Yeah, that's probably the neocortex, and probably the rest of the brain is like that, but with more mistakes, with lower quality, perhaps. So my idea that's in the neocortex, the signaling that determines the tiling, the signaling that I think are repulsive pathways like heparins or semaphorin, probably is more developed and it works better. But in the rest of the brain, that occurs, but perhaps in a more primitive fashion, more taking a picture in a more primitive fashion. But
everything.
So probably the same elements all over the brain, but with different quality.
Okay, so the astrocytes, they have this really dense projective field of processes, which is, you said, about 50 micron across?
The whole size, yeah. Okay. So the ball is the diameter, up to 50 microns.
Okay.
The whole ball. From 35 to 50. But 50 is a good number.
So would that be compatible to something like a cortical column?
I think columns aren't they larger? I'm not sure about this. And they're wider. I think they're wider than that.
But do you see it aligned with, let's say,
I think
the structures that neurons?
No, there's one paper where they do fate analysis, so they label the astrocytes and they see in the adults where they are with radial glia, and they do see columns, but they are not all over the place. So it's not that columns have astrocytes associated to that column, all the columns. The notion is that some of the columns have astrocytes, indicating that the origin of astrocytes is mixed. Some of them come from the radial glia that gives rise to the column, and some of them comes from elsewhere. And that creates a complex pattern.
And the astrocytes would only be in the gray matter of the nervous system?
No, there are also white matter astrocytes. Absolutely. They're different as well, but yes, they are. And they have a different morphology and different molecular phenotype too.
But it is essentially just a continuous matrix, if you want, of astrocytes.
Yes.
So if I take a brain, if I could visualize all the astrocytes in that brain, it would be just the whole volume would be covered with equal, or sort of very regularly spaced
probably
astrocytes.
Yeah, absolutely. Yeah. With the different densities, different morphologies, depending on the area, but yeah.
But it will be a continuous.
Yeah, that's a nice experiment to do with the clear brain technique.
Exactly.
Absolutely, Bori, you were thinking about that? Yeah, that's a experiment.
But that will be the prediction.
To stain astrocytes exclusively, and we will see different tiling of astrocytes all over the brain. Yeah, absolutely. That would be nice.
So
a nice picture to have.
Would that structure then? So what's the density then? If you take just a bit of brain, a cubic millimeter, right? In your mind, then these mushy astrocytes would really fill up that whole volume, and then the neurons and the blood vessels and others are sort of squeezed inside that matrix, or
everything is a squeeze there.
Yeah.
And probably the arrangement of all the cell types is highly coordinated and regulating, and vessels probably play a big role too. Right. So the vessels grow and everything adapts to the rest of the space and creates their own circles there.
But then couldn't you speculate that these astrocytes are there to give some sort of uniform mechanical support
That's interesting
to the rest
idea.
of the system, right?
That could be it. So I think that when astrocytes were described as a glue, initially named glia, glue is not a bad term for astrocytes actually, in the sense that they're all over the place. The difference is that glue implies is a passive support, and this is possible. Perhaps there is some, as you say, structural support for the rest of the structures. But I do think myself that structure is also actively exchanging information with the rest of the cells. So it's not just a passive thing there that you may have break or real glue. It's an active glue. So whether it has also supportive elements and guiding elements to its structures, it is possible.
Yeah, because would you know of any other cell type, non-neuronal or neuronal, that would have this feature of such a uniform distribution locally and globally, right? And
I think microglia too. Microglia is all over the place. Exactly. And
also like a three-dimensional matrix.
Yeah. And NG2 cells, which I find fascinating. And we don't know much about them, but they are all over the place. They're highly abundant. And they're also, they have a tile. Microglia and NG2, they are also tiled. They're territorial. It's just neurons that are not territorial, but yes, they are. And then if you could stain them, you actually see them beautifully arranged, regularly arranged all over the brain.
Right.
Absolutely.
So in some sense, you want to have a very strongly symmetric structure to distribute all mechanical forces so that you can allow neurons to be, let's say, asymmetric in how they
That's an interesting, this could be an interesting idea.
Okay.
You think like an engineer.
I don't know. Do I?
That's an engineer thought, but why not?
I
think myself about, for me, the tiling, it's about information processing. So
that's indeed the next question, right?
For me, that's the idea. So they segregate information processing in a set of neurons. Either they synchronize that activity or they provide energy for a given set of neurons. So for me, that's the notion. It is related to information processing. Like in a circuit, it's a mini circuit. And I have no idea what they do there, but tiling is more associated to synchronization of activities in a given module in the brain.
Okay, so let's look now at function, right? So here we have these astrocytes, equally tessellated. We don't know what they do, and it's actually a significant chunk of the cell volume of the brain.
That's correct.
So you spoke of the dark matter of the brain. And now we can start to speculate about what they do, because actually we don't know much about that. No? So why has it taken us so long to even start to worry about what they might do functionally? Why?
No, that's a great question. I realize, and actually this is sort of a recent understanding about things. Research develops very, very slowly, incredibly slowly, which is for the people that are not very patient, that includes me. That's very striking. So it takes us years to find something relevant. And sometimes the techniques are a limitation or an advance. So in the case of astrocytes, we spoke about this morning. The reason why we know so much about calcium astrocytes because we have calcium imaging. And that was fantastic. And since we don't have imaging or another readout for another phenomenon that may be even more relevant, then we cannot talk about that phenomenon. So sometimes techniques determine concepts. So we have to be very aware of that. So as to why it has taken so long, I think a big problem is, in addition to science being slow in nature, that the neuronal people haven't been interested in non-neuronal cells. Mm-hmm. Even if we look at the number of references, I have a student, my student Abel, that just defended his thesis. He had a picture about the number of citations, so the number of articles related to astrocytes, the number of articles related to glia. There per year. So we have like 20,000 articles for neurons versus, I don't remember, 2,000 articles for astrocytes. Mm-hmm. So then that explains a lot. So the number of people working on neuron is larger than the number of people working on astrocytes. That determines also how fast discoveries goes. And the second thing is that neuronal people are not interested in astrocytes. Mm-hmm. That's it. And then many of the neurons, the advance in neurons is earlier because the action potentials were discovered in the 50s. Advancement in astrocytes started to happen in the 90s mm-hmm. when we discovered calcium imaging for astrocytes. So we have been studying neurons for 50 years already. By the time we start to realize astrocytes do something else other than being a glue. So that can explain a lot of things. The techniques are different. But I think a big problem is that neuronal people are not interested in non-neuronal cells.
But conversely, you could also the astrocyte people don't care too much about the neuron.
Yeah, exactly. But that the impact of that in the neuronal field is minimal. I think the impact of all these people working on neurons of not caring about astrocytes is much larger.
Yeah, but I think that's a very common impression people have, that whatever they do is not considered relevant by their colleagues. So
that's true.
I think it's just a sign of general fragmentation.
Well, but in our case, we have numbers. So systems neuroscience concerns mostly neurons. Mm-hmm. So if you in the big meetings that are specialized in system neurosciences, we calculated that only 1% of presentations are dedicated to non-neuronal cells.
Yeah, but maybe the problem here is that right now there's not a clear proposition what astrocytes could contribute functionally to neural systems. Exactly. Right? And I think this is the big challenge. And in some sense, if you say 2,000 papers a year on astrocytes, then you can also wonder, okay, what the hell are these papers about, given that our overall understanding is still so limited.
So the question is how astrocytes contribute to higher brain function.
That should be our challenge.
That's our challenge. Yeah. Beyond clearing potassium, which is very important, or beyond providing fuel to neurons, which is very important too. But it did do something else that is relevant to higher brain functions. And if we didn't have astrocytes, we wouldn't have those functions.
Mm-hmm. No, so let's take a look. What do we know about the interaction between astrocytes and neurons? What are the outstanding features that astrocytes have that might be relevant if you are a neuron?
Let's go for the data. So the data shows that you manipulate astrocytes and you change the response of neurons. So before any further interpretation of why astrocytes are necessary for neurons. So in local circuits or in networks, doing things to astrocytes, manipulating their responses, changes the circuit readout, changes the network readout. That's a fact. That's the evidence, the experimental evidence. Now,
then I could say, look, if I just occlude the blood vessel, also something happens to neurons, but it's not necessarily
one thing is that it's different if neurons suffer because they lack like basic support, they lack oxygen. And another thing is that the neurons don't have action potentials, or they have reduced action potentials because astrocytes are releasing different amount of transmitters. So then the signaling that is involved there is very different.
Yeah, but for instance, what I was looking for, as you also discussed, there's direct exchange of key ions that neurons need to be active, to change their membrane potentials, to generate action. But like there's sodium exchange, you have potassium exchange, there's a calcium exchange between neurons and astrocytes, right? So apparently these ions are exchanging. So what are astrocytes doing with it? What is our astrocytes doing with potassium? Are they buffering it? Are they
buffer
diffusing it? Are they?
They buffer it. They bring it out of the extracellular space to the blood. That's the standard knowledge. They do have a lot of potassium channels. And in diseases, it's well known that it's a good therapeutic target in the sense that they get dysregulated, meaning that probably if they don't buffer potassium correctly, that contributes to disease and to neuronal activity.
So the interpretation goes again into a more homeostatic direction, like, okay, it's sort of just removing the potassium from extracellular space.
Right. But another interpretation, it's like basic computation. So the fact that we spoke about it, filtering, so if there is a circuit, imagine there is a circuit and neurons are part of the circuit and astrocytes are part of the circuit as another element. So they may be doing things to that circuit.
That's what I'm trying to figure out.
Like they're maybe gating, changing gain, changing extras on the
could they release potassium again in extracellular space?
They could release potassium, they could release glutamate that goes back. It's like a third neuron that goes back to the neuron and changes the way the neuron responds to the next stimuli.
So it means that the astrocyte essentially can control whether a neuron will respond at all to the next stimulus.
It will modulate. So I think it's better.
In the extreme case, it could actually prevent it.
Yeah, exactly.
Yeah. And
even could change. So it can. There in the studies in the local circuits, so it is known that depending on the input, so the inhibitory neurons, for instance, depending on the kind of response they have, they produce different responses in astrocytes that in turn relay different outputs to excitatory neurons. That's well known, and that's pretty delicate. So it's not just homeostasis. It is pretty balanced reading of the neuronal activity and routing it to the other neuron in the circuit. So that for me, this is beyond homeostasis. Mm-hmm. This is the astrocytes acting like another neuron, right. And they can act. They can make change actually the output, the global output of a given circuit can be changed by astrocytes.
Mm-hmm. But now,
in a precise manner, beyond
exactly.
Not beyond like vascular cells delivering oxygen to neurons. But they can change that. For you, that doesn't mean that astrocytes may contribute to higher brain functions beyond just their metabolic roles.
So look, I think it would be great if astrocytes would help out. So I have no objections against that. So it's more about looking what they can really do and about spatial temporal scale that could play out.
That's a good question.
If we talk about the potassium dynamics, this could play out at, let's say, millisecond level, the buffering and release of potassium? Or do you see this as a slow process?
I think it's more a slow process, but I have to look into it.
And glutamate?
The glutamate uptake, I think it goes in the millisecond scale.
Mm-hmm.
Glutamate release, it's in the second scale. So when the astrocytes respond to neurons by releasing glutamate, that takes seconds. Mm-hmm. But in hundreds of milliseconds and then in the tens of seconds.
And this release is then non-specific in extracellular space, or is it very targeted?
It's sort of paracrine. Let's say paracrine. which is not a bad term because there are neurons like the noradrenergic neurons that work in a paracrine manner in volume transmission. So sometimes relevant things happen globally, not just locally. And probably local control is different from global control.
Sure.
Both have different roles.
So you're saying there's no evidence that astrocytes would have the specificity to have a very highly temporally precise and locally specific.
No, it's more
global.
It's more akin to volume transmission, I think. I think that I see astrocytes in the anatomy, the way they relate to neurons, their time scale supports global roles more than very local roles. So the point is to look what kind of global roles, synchronization of different neurons at the same time, are relevant for astrocytes to control.
Now we talk about different neurotransmitters, and for some potassium, are these only ionotropic receptors that astrocytes have, or do you also see metabotropic receptors? Where if you look at straight at
they have metabotropic receptors for glutamate, for instance. They are typically present in astrocytes. Yeah.
Doesn't it make you suspicious that there's actually a real functional role for this? Because that would mean that you really actively, in a very specific way, are regulating processes in the astrocytes depending on the presence of glutamate.
Oh, absolutely. Absolutely.
So this would be also a little bit beyond just buffering, no?
No, absolutely no. Yes, even transport of glutamate signals to some functions, even just the mere transport.
So what kind of reactions do you see in astrocytes to glutamate?
You may have changes, for instance, in the physical changes in the way the astrocytes wrap neuron. So the same way that spines change their morphology in order to get closer to another spine, to retract, that has been described also in astrocytes, in those distal processes in relationship with neuron, and this is regulated by glutamate and calcium.
So you would argue that the glutamate then also regulates, if you want, the formation of these,
for instance,
of synaptic structures that would then pre-postsynaptic neural
exactly, and then
plus your astrocyte.
Yes, and an area of astrocytes that I'm very interested in and it hasn't been explored are long-term changes. So we know that astrocytes modulate memory because when we manipulate the astrocytes with chemogenetics or transgenic tools at different stages, now has been done with chemogenetics at different stages in the memory paradigm, from acquisition, consolidation, replay, so that exactly where they're acting is getting characterized. But we don't know exactly what are the long-term changes in astrocytes, the same way that neurons have long-term potentials, LTP, which causes changes in the molecular makeup of the neurons. That hasn't been characterized in astrocytes.
Oh.
And I do think this is an area that I'm very interested, because I think that can be very fruitful to understand.
Right? Because, for instance, there's this whole problem at the neuron side. If I build a synapse, how do I stabilize my synapse? And there's the idea that this also has to do with intracellular processes that depend on other molecules, like chem kinase 2, as an example, right? And John Lissman hypothesis. But that would suggest that you need similar kind of memory processes at the level of the astrocyte if they form part of that synapse, right?
Yes.
So is there anything known about these kind of intracellular signaling pathways in astrocytes?
No, not at all.
Okay.
That's a totally infant area of study.
So you study one of these 2,000 papers a year are all about, right?
Yes.
But then the other possible control that astrocytes have is capillaries.
Absolutely.
Right? They control.
Absolutely.
They can directly control blood flow and exchange, right?
They
also
do.
The blood-brain barrier. So how do you think that can be or is used, and on what time scale is it used by astrocytes?
I think, that's what the data shows in the time of milliseconds. Absolutely, that happens. So when a neuron in somatosensory stimulations, a neuron in mice, the whiskers touch the neuron, detects that you have action potentials and calcium inactivation. Then milliseconds later, hundreds of milliseconds, astrocytes see it, and they transfuse that to the vessel.
Mm-hmm.
That's a fact.
Mm-hmm.
And it's interesting when we talk about this morning that they could do this earlier before the task happens, because there is a prediction that is going to happen, and so it prepares the territory for a better response later on. But yeah, absolutely. The relationship of astrocytes and vessels, for me, is very important. It's very, very clear. And I think astrocytes multiplex, so they can talk, and structurally they are different because the distal processes are one thing, and the anther is another morphological structures that is different and wraps the vessels, and it doesn't have the bulbous, very dense network and these lattice in the distal process. It's just like it's like a sheet, totally, that wraps the vessels. So the astrocytes have the multiplex. They're able to multitask, and one task is regulating the connection between vessels and the rest of the brain.
But
absolutely.
What's the delay? So when we have neural activity, with what delay do the astrocytes start to then change a blood vessel response, and with what specificity?
In hundreds of milliseconds.
And
hundreds of milliseconds.
Okay. And that's a long lasting. How long does this?
I would say seconds. I'm not sure about that, but seconds.
Okay. And then I would assume that this is not, if you have one spike, I don't think the astrocyte will start to change the dilation of the capillary.
Oh, that you need
is there some
threshold? I don't know about it, but probably.
So would you see then the astrocyte looking at some average neural response, and given this average, it would control the capillary?
That's a great question. I think it's none.
Okay.
Whether the response to, because usually the experiments are more one-to-one, but I don't think that has been analyzed, whether you need a minimum of neurons to be stimulated in order to produce.
Now, one reason I'm asking is, of course, a lot of the work on magnetic resonance imaging and how you can use that to assess function in the brain is by looking at this bolt signal that's derived from blood flow. Right. And then there's a belief that this is related to neural activity. But now we see that this is actually mediated by astrocytes.
Well, it could be mediated by astrocytes in the sense that astrocytes consume oxygen. That's the point. But not all the time. So it's not that thing. Well, both signaling is actually very complicated, because what it really shows that is there is hemodynamic response. So it is an increase in blood flow, but actually oxygen consumption doesn't increase. It's the paradox of both that was described several years ago. So meaning that oxygen consumption doesn't change very much. What changes is that there is more blood flow. And as a result, the ratio of hemoglobin that is oxidized versus the hemoglobin that is not oxidized changes. This is what you're looking. You're looking at increasing flow. You're not looking at increasing oxygen consumption. Both is really difficult to interpret. But in general, it has been used as a surrogate for neuronal activation. And this is what I think is wrong. Not totally wrong. It's just neurons can contribute to the signal too. Not all the time. So my point is that we need to get into the details. I think the paradigm is very relevant, because the notion that the task-dependent activity is the model of brain activity is wrong. So we need to look into different models, including that one of high glutamatergic activation, but also intrinsic brain activity. I'm sure that the both signaling intrinsic brain activity is very different. Gustavo Lecoq works on that. From task-dependent activity, changes during the day, changes in different nuclei. So we need to get out of average measurements and very specific models that we think are the models of all the brain activity, because the devil's in the details here.
Right. Exactly.
And I'm sure that in in very specific circuits and in very specific moments, astrocytes consume a lot of oxygen.
There are some papers in the literature where people look at this relationship between neuroactivity, astrocyte response, and blood flow. And that are mainly done with optogenetics, and they're being questioned whether they actually tell us whether they're informative. On the other hand, you can also understand people question it because for many neuroscientists, there's a lot at stake to just believe in the dogma that, the bold signal reflects neural activity.
Right.
And so, but what is the truth of the matter in your view? Is it like the neural contribution will be like a tiny fraction of the bold signal, let's say 10%, or it will strongly vary on conditions, and so because it's high variability, it's uninterpretable. So where do you?
I think that the right measurements need to be done. I wouldn't be able to calculate, and this is like the traditional biophysics and the brain physiology. So we need to really calculate that.
But you could also use.
But I wouldn't dare to say that astrocytes use more oxygen than neurons or vice versa. So it is clear that action potentials are very demanding of energy. That's absolutely very clear. The needs, energetic needs of astrocytes are not known, for instance. We don't know. There are pathways and there are phenomena in astrocytes that require ATP, very many, exocytosis, calcium removal. So there are many pumps in the astrocytes. Perhaps we can say that they don't use as much as ATP as neurons, but I wouldn't dare to estimate unless I do the calculations. So the energy requirements of astrocytes are not known at all.
You said measurable for you?
I think they could be measurable. Yeah, I think this is like they have been measured in neurons. So why not? And the thing that we know now is that at least they are able to produce ATP through fatty acid oxidation. That we think they do, which is very efficient to produce a lot of ATP. But we think that they don't have fatty oxidation all the time. We think they're versatile. The beauty of astrocytes, if I can introduce this word, beauty, in a scientific talk, is that they're versatile. Mm-hmm. So they use whatever they have, and they have glycolysis. And probably because the ATP produced by glycolysis is necessary for certain pathways that are locally by the membrane and related to the release of glutamate or to uptake of glutamate. But they do generate a lot of ATP through fatty acid oxidation at some points in their life. I'm not sure when, because we haven't carried out experiments in vivo, but I'm sure they do. Mm-hmm. And in certain moments, but I wouldn't dare to say whether that accounts for what percentage of brain oxygen.
Astrocytes can operate in an energetically efficient regime, but they can also be energetically inefficient. It can be both. You say at some point in their life.
I think that they can use only glycolysis, and what happens there is they don't die. But one thing is not to die, another thing is to be doing well.
To be useful.
To be performing right. One thing is survival, another thing is the right performance.
No, it's interesting because it again would argue against this homeostasis interpretation, because if it was homeostasis, you would expect that substrate to be very energetically efficient, because otherwise how can you be usefully contributing to maintaining homeostasis of your neurons if you burn a lot of energy yourself?
That's actually
and
correct.
Produce waste products and so on. Right?
I think we need to understand the energy metabolism in astrocytes, not just when they use what pathway in what moment. Also, I think this notion that ATP is not stored perhaps is not true. So there is something that is very labile, you produce ATP, and then either you don't use it, it's gone. So perhaps astrocytes store ATP too, so that allows them to resort to glycolysis when neurons need a lot of oxygen. Then within the oxygen goes to neurons, not to astrocytes. Right. But the astrocytes are still there doing something, so perhaps they can use ATP from other sources. So there are questions there that need to be answered. Right. And I do believe that energy is very, very important, and that many of the anatomical designs in nature are determined by energy usage and energy usage optimization.
Right.
So that could be probably a very fruitful area of research to try to understand how astrocytes, the interplay of energy metabolism between astrocytes and neurons, how much oxygen each one uses. And
so is there any evidence that astrocytes can really, let's say, turn circuits on and off, and then it also happens under realistic conditions?
I would, well, experimentally, I have to find the papers. I would say yes. So probably if you ask this question to Alfonso Araque, or leaders in the field, yeah, I think they can actually
So that means a sort of energy management.
Because of the energy, but also because they stop releasing a given gliotransmitter that neurons. Yeah. Yes, I think they can. Yeah. Yes.
Right. Okay. So
not just because of the energy, but because of the signaling.
Right.
Yeah.
But on the other thing that I always find very exciting about astrocytes, also because I'm ignorant, are the gap junctions, right? So they have gap junctions that allows potassium to just permeate across membranes between astrocytes, and that would, in theory, set up possibilities for more rapid signal transduction than what you would get if you have action potentials going over axons. Would you agree with that?
I'm not sure about that because the unit is not. So the potassium that is diffused out of the astrocytes would have reached enough concentration to change the neuronal activity in other neurons that are farther away.
No, let's just worry about the network of astrocytes and they're coupled with these gap junctions, right?
Right, so but then in order to have an effect on neurons, you need to have really reached certain levels of potassium. So I tend to think about diffusion as not being very controlled. It's just passive. They are removed. So I'm not.
It depends on the gradients, right?
Right, exactly. But that has to be modeled. So the answer could be.
Yeah, but look, if neurons are very active, they are transiently buffering a lot of potassium, right? So that could possibly set up these kinds of gradients. No, because then you would have then the potassium in the astrocyte network would be rapidly attracted to these areas where there is activity.
But a gradient for me is a process that is regulated in a fine manner, and this is incompatible for me with diffusion. Isn't that incompatible? So I tend to think that diffusion is just
together, no?
Yeah.
So
but that could be modeled. I think that idea could be put into a model.
Absolutely.
And tested different levels of potassium production and potassium removal by astrocytes, and to see that creates gradients.
Yeah.
And those gradients change neural activity. Yes. Maybe but you mentioned about the priming of neural activity. Maybe this is created by gradients of potassium around a given circuit that makes the neurons respond more or less, or beyond a threshold, or not reaching the threshold. So maybe but that can be modeled.
Yeah, because what you would have, let's say you have neurons that become very active, so that they start to buffer a lot of potassium rapidly. This might then deprive their neighbors from also becoming active because there's no potassium around. And then the astrocytes allow potassium to flow in from other parts of the network that are, let's say, so in some sense, it also would give you a competitive system, because if I have different islands of activity, they, via the astrocytes, can start to compete over the potassium.
I see.
Right? And then this could play out very quickly if the gradients are big enough. So would you buy this kind of fantasy scenario, or you think this is really completely?
I would like to see the models.
Okay.
I would like to see the models that support that kind of scenario.
Okay, because the same thing might also be then going on with your sodium. Right?
Exactly.
But the sodium doesn't diffuse over the gap junctions, right? That stays local in the astrocyte.
Right, it's taken out by astrocytes. It's true that it's taken out by astrocytes, yes.
So it's only the potassium that could do this.
Yes.
So the other thing I was thinking about that is exciting about these potassium networks or in the communication channels across the astrocyte network is it might give you a channel to start to prime different areas of the brain, right? So you mentioned this also earlier where you say, well, we expect a vision, and there are experiments that also have shown already that you have anticipatory changes in the Volt signal that do not correlate to neural activity. And this would suggest that this is driven by the astrocytes. But then the question, how can astrocytes know? Right? So there must be some communication channel that says, look, we expect a visual stimulus, so increased blood flow, whatever.
Well, but this is also the prediction brain, that the brain has already the information about the outer world and is renewing that information all the time. And whenever new information comes in, they just reset. Then, and perhaps astrocytes are already in that circuit, meaning that they are actively processing information all the time. So premonition is part of the system that is replaying the information of the time. Don't you agree with that?
Well, I was more thinking about that you just have, let's say, a conditional event that you're conditioned. When you hear a sound, there will be visual stimulus 5 seconds later. So I hear the sound, and now in an anticipatory response to this association, I start to prime my visual area through the astrocytes to say, okay, we're going to get a visual stimulus, wake up. So you can sort of put areas in idle mode, like low energy consumption, and then reactivate them from that by just through the astrocyte network.
It could be, yeah.
Is there any data that would support that idea?
No.
Except these anticipatory bolt response in the visual cortex, right? Was that Crutchfield or?
I don't know. I don't remember. Yeah.
Okay. So now, so
it could be one of the reasons for tiling. So that creates different islands of energy usage or potassium gradients along the brain. Yeah, could be.
Could I, if I'm an astrocyte network, could I control the diffusion of the potassium? Can I open and close my gap junctions? Are they actively controlled or are they just passive?
Nothing, they're passive. Okay. Yes.
So you also, so now we made a little index of different possible functions of the astrocytes. And you also mentioned this lymphatic system. So, and that of course pushes us a little bit more in the direction of homeostasis.
Absolutely, that's homeostasis, clearly.
So why was this lymphatic system relevant in this discussion? Why do you think this is an important insight in terms of what astrocytes could be doing?
Well, because it's I think a very important issue as to how the brain removes waste. And this is highly, highly relevant for normal function. It is highly relevant for diseases. So I think it is worth mentioning. And it's a very recent discovery, so it's worth mentioning. And moreover, it is probably regulated by the brain itself. Mm-hmm. So it's not really clear at this point, but the locus ceruleus and some of these nuclei that are globally broadcasting throughout the brain probably regulate that too. And there are studies showing that this can regulate also. You spoke about gradients of potassium determining circuit activation. There are studies showing that. Other studies showing that these fluxes inside the brain control the glucose availability, the availability of nutrients. So they're not trivial. They're not just things that are there. They're very relevant to brain physiology.
So the astrocytes are sort of regulating blood-brain barrier. This sets up a convective flow of what?
Of liquid, it's liquid that moves from the artery side to the venous side. It's just intercellular space.
But what's the liquid? What does it?
It says solutes, salts probably. It's convection. It's pure convection, and it's probably water.
So it's really like you're washing out the brain.
You're washing out the brain. Absolutely.
Where does it flow towards? Is it Where?
It flows to the venous side that eventually goes to the lymphatic system.
Okay. So it's really like...
It's washing it's washing it. It's actually beautiful physiology. I'm very fond myself, in addition to computation, to brain physiology studies. There are, and these studies actually very elegant because it's actually very simple. Technically, they use fluorescent tracers and they just put them one side and see whether they can find them. And then they find their way out of the brain and they can track the pathway. So they absolutely, there is water, the brain is cleaned every day. With a lot of things get out. And that is mediated by the astrocytes in the sense that they are a wall. At least they're a wall that those things have to cross. And that wall probably is also selectively regulating the passage. And when that wall is not working well, then that passage doesn't occur. And that changes also during day and night. I find it fascinating. The finding that this clearance works only at night is fantastic, meaning that the intracellular volume of the brain changes during the day and during the night.
Right.
So sure, if the astrocytes shrink or something, and that changes the convection towards the venous side of the brain.
What distances are covered in that way, you think?
I'm not sure.
So what's the density of this venous network?
I'm not sure, but probably that answer can be found in the literature. Yeah. But it's fantastic. But
it also means that there must be different chambers along the artery, no? Because you must accumulate this fluid that's being released. It must be extracted from the blood, stored, and then released.
Well, it comes from the CSF. So the CSF is in contact with that liquid. But that's all physiology is well known. It comes from the CSF. The CSF is circulating all the time and is in contact with the intracellular space.
Yeah.
Probably they have different contents, but there is a flow. There is a flow from the portal in between the arteries and the astrocytes that goes in a different direction from the blood. This is interesting also. So the blood goes in one direction and that flow goes in the opposite direction. And eventually, because of these convection forces, crosses the parenchyma to the venous side.
But the astrocytes must be controlling both walls.
Yeah, they do. Absolutely. Yeah.
So they must be controlling the artery wall.
Absolutely.
Right. And then this CSF chamber.
Yes, absolutely.
Okay. Yes. But so do you know when in sleep this happens? Is it like in which sleep stage does it happen? Do we know that?
I'm not sure. I'm sure they have looked at it, but I don't have the data. But as it occurs, the paper has very beautiful pictures and it's very dramatic. The way that those fluorescent tracers go through the brain at night, but they don't go through the brain during the day. In mice, it's really, really dramatic, the change.
Right. But so now at the more behavioral level, you highlighted a number of correlations we are aware of with respect to astrocyte activity and behavior, like in sensor processing, state switching, and fear response regulation, right? So which of these results for you are most informative about the possible functional role of astrocytes?
Brain states, I think network activity regulation. Because of this global notion, I think the findings in the local circuits are just a part of a larger role. So they find them because they look, but I think the astrocytes are more important in the regulation of networks. So they are more relevant to these brain state transitions. That's my, I think if I would study something, I would study the role of astrocytes in neuromodulation. So global phenomena, neuromodulation, brain state, slow oscillations. That would be for me the.
So in the brain state example, you have use optogenetics to stimulate
the astrocytes, Kira post-cancer in Kira post-cancer, the paper.
Yeah, right, exactly.
And Rafael Justin and Kira.
Right, exactly. But you're stimulating in which area are we? It's the somatosensory cortex, or was it the barrel cortex?
I think it's the somatosensory cortex. Yeah, it's the cortex.
Okay. So we're measuring LFPs, and what we then see is that after a 5-second stimulation, you essentially see a peak at about 100 seconds later of really low frequency, like below, let's say here, 0.5 to 2 hertz, or really in delta range, right?
Right.
Which is a frequency range that under the control condition does not appear, right? So this here.
Right, there's a change in the power.
So
at the low frequency that is increased and stimulated.
Like there's actually no power in the control condition.
Exactly.
And it jumps up when you stimulate the astrocytes with optogenetics, right?
Exactly.
But now, isn't that a surprise? That's no surprise, because if you drive the astrocytes stimulate astrocytes for that long, you might lead to, let's say, pathological change in the local
But
volume.
not quite pathological. I wouldn't say pathological. That it's not known. So
low delta would correlate with really deep sleep or deep anesthesia, right? So it also would mean these neurons are deprived of just any ion to do anything.
That's a good
so
thing.
essentially shutting down. So this is like you have just shut down the neurons by depleting them of any resource.
What would be the right control to use?
Well, if you want to see state change, you want to look at oscillatory frequencies that you know have a functional relevance, right? So you want to look at frequencies that's either in gamma or in theta or alpha, but certainly above delta, because delta would reflect deep sleep. So my question here is, can we really speak of a state that is functionally relevant? So you have to look at a frequency range that you know has functional relevance.
Right. This finding has been reproduced. Actually, it is an early study by people have shown that if we remove the capacity of astrocytes to respond to the nucleus basalis, the nucleus basalis are minor. They also regulate brain state. And this has been measured with local field potentials.
It's very different.
But the result is very similar. In this case, they are not stimulating aspects with optogenetics, because optogenetics didn't exist. I think we're talking about a study from 2000 something. I don't remember the year. But the result is similar in the sense that if the astrocytes don't have calcium responses, the nucleus basalis minor doesn't regulate the brain states.
Mm-hmm.
That's
nucleus basalis minor would be the cholinergic projection,
right? But meaning talking about so in that case we cannot talk about astrocyte stimulation depleting neurons from things they need from shedding, because it's just the opposite experiments. Astrocytes are not being stimulated; they just don't respond. Therefore, they cannot be depleting neurons from anything. And it's the same result in the sense that they regulate brain state.
But now in this optogenetic experiment, what does it mean to activate an astrocyte? Because they're genetically manipulated to respond to light. Right?
That's a key.
What do they do?
That's a key question.
How are they coupled? To which receptor is it coupled?
It's not known. It's not. That's a key question. That's the finding. They express archirrhodopsin there. They stimulate with light, and they see that calcium increases. But how it happens?
Calcium inside the astrocyte increases.
The calcium transients inside the astrocyte increase, and the only thing that they know is that archirrhodopsin hyperpolarizes cells, and in neurons that triggers the neurons are inhibited. In astrocytes, there are also voltage dependent channels, but not as active as in neurons.
Okay.
So probably the astrocytes are detecting the hyperpolarization somehow, and that triggers somehow an increase in calcium concentration in the cytosol, but it's not known how. First thing. Second thing, it happens mostly in the processes. So it's a very important thing whether the calcium is increases are happening in processes, in the distal processes or in the primary process or in the soma, because the mechanisms that regulate calcium release are very different in different compartments. And interestingly enough, with archaeorhodopsin, they happen mostly in the processes, indicating that those voltage-dependent channels are perhaps richer there, but it's not known. It's not known. And I think this is a limitation. It's very interesting because it's actually interesting to be able to stimulate astrocytes or neurons any cell type in a time-specific manner, and not using transgenic mice that are... It's a really elegant tool, but the fact that we don't know exactly how it works, I think is a limitation.
Right.
Because we don't know the physiological relevance of the response.
Right.
It is a limitation.
This is a bit problematic then, right? Because we can speak of a state change, but we don't know whether we have induced some pathological state of the tissue.
Understood. I mean, we don't know.
It's also sort of not sufficiently controlled.
It's probably we need to refine the technique, understand mechanisms better, and find the same things using other approaches.
So we're still in the dark then about...
We're in the dark.
What it does, right? But now, in your case, you are also interested in finding... there's another problem we see here. Everything is so much tied to calcium imaging, right? In astrocytes study.
Too much, probably, yes.
And this, of course, is also biasing very much
how
we think about it, because we think, these guys are really slow, right?
Right, exactly.
But this might be due to the way we observe them. So would you expect, if we would have the magic imaging techniques, or measurement techniques to assess the dynamics of
electrophysiologically like techniques.
Right. Yeah. Do you expect them to be as fast as neurons in terms of time constants at work, or do you would your prediction be that they are a system that are slower than neurons?
I would say they're slower. Perhaps they are faster than seconds. So maybe we can move now the time scale from hundreds of milliseconds to tens of milliseconds. Mm-hmm. But still, that's slower than neurons.
Okay.
Right. I would say they're slower. And my point is that that we have to find the moments where the brain needs that time scale. Mm-hmm. So we don't need to make astrocytes faster than they are. That's a mistake.
Right.
So the field has been trying for a very long time to make astrocytes look like neurons. And that's a mistake. We just have to find what they are. And once we characterize them without forcing the interpretations, try to find out where that time scale is relevant for. Is it relevant to any phenomenon in the brain? I think this is the way to go. Not just these very ultra fast representations of neurons that last few milliseconds, that astrocytes have nothing to do with it, obviously, because that's beyond their time scale. But other phenomena in the brain that happen there, where the astrocytes can be useful. That's, I think, the way to go.
Okay. So now, could you imagine astrocytes as a substrate of memory? Like his memory is to carry information over time, and you do that by stable structural change. Do you think astrocytes have the properties, the basic also biomechanical and the biochemical tools to form memories and store information over time?
I think it's a biophysical question whether they have something that can have many different positions to store information. So that's the question. And the question is, can different calcium transients do that? Mm-hmm. We don't know. We have to ask that question and analyze it.
Because the question is, of course, then, if they provide context in which neurons operate, this context might have to also be associative.
Maybe. Right. The question is whether this context is just supportive, metabolically supportive, or structurally supportive, or it's really an intelligent support and it's doing something for neurons. Mm-hmm. There are the questions of what are the variables that neurons encode is not totally clarified, because they know that some variables like odor or sounds or colors, I know the neurons encode those variables and in representations that are happening in the time scale of milliseconds. But there are other variables that are for instance, there are many other variables that the brain uses, and I don't think we have discovered them, and perhaps some of those variables are bolder, or and that is the realm of astrocytes. But that are very important too, like associations, contests, providing averages. Perhaps they do that. They carry out the statistics of the brain in this model of prediction coding, where the brain is so tied doing the statistics about how their predictions are confirmed by experience. But that's a lot of error calculation. And some of, I've reading papers that describe that some of the parameters around the error calculation take place in a time scale of seconds, actually. Some of them are really fast, and some of them are slower, the ones that require more computation. And that perhaps is this kind of buffering.
Well, probably
is perhaps what the kind of things that astrocytes do. So they're not, I don't think they encode for concepts or for colors or for others. I think those neurons do that, and that is related to their to the lack of specialization of astrocytes. So neurons are highly specialized. And for me, that means that they encode for many different things, place and music. I don't think astrocytes do that. I think astrocytes do general things, general computations, providing general context in a time scale of seconds. So the question is, what variables are encoding? That's the question. But I think there are different variables. They're broader, but those variables are probably important too in determining.
One way to get a handle on that is, of course, we could look at the different pathologies where astrocytes are involved. So what are the outstanding pathologies?
They're involved in all pathologies. Any brain pathology has sick astrocytes. The question there, so they're morphologically changed and molecular change. The question is, what is the contribution of that astrocyte to the pathology?
How many diseases are there that specifically attack?
There are, yes, Alexander's disease. There are some diseases where GFAP has mutations, and that causes changes. Yeah.
So what are the specific phenotypes you get?
They are retarded. The children that have the problems with the blood-brain barrier. Yes.
But are there any diseases that specifically detect astrocytes later in life, in the adult stage?
It's not known. Mm-hmm. It's not known because astrocytes also get older, and probably they are dysfunctional, but we don't know, and they are affected by disease. The question is whether this is that an epiphenomenon, whether it's just the main cause of the disease, or it's just a lateral thing. That's the question, but they don't work that well.
But you are interested in developing therapeutics.
Yes, because we have data, and we are very driven by data. Then when we manipulate astrocytes, we manage to recover brain function very effectively.
Mm-hmm. And that's in Alzheimer's disease.
In traumatic brain injury in Alzheimer's disease, we are still trying to understand the molecular makeup of the astrocyte change, because the problem is that we cannot, so far, we don't have astrocytes isolated from human brains for obvious reasons. So we are trying to understand what happens to astrocytes in Alzheimer's disease. Mm-hmm. Happens means all these homeostatic functions, metabolic functions, are they changed? Mm-hmm. And if they are, how does it contribute to disease progression or not? Does it make any difference whether we treat them or not? So my understanding is that we should go for combination therapies, and focusing on the neurons is a big mistake, because going back to the vascular system, even if you have the finest treatment for neurons in order to help them make these amazing spines, if the blood system, the vascular system is not working, the treatment is not going to work. So it's a very obvious thing, but the field is not realizing that some very obvious things are not being done. And for me, recovering the function of astrocytes, it's as important as recovering the function of blood vessels in order to cure any disease. And in addition, perhaps then we can target the neurons more specifically, but I think recovering astrocytes will have a major impact on disease recovery because of the many things they do.
Right. So that's a fantastic outlook, right? So in your career, you have, in some sense, you're in this minority field, right? That is not necessarily in the limelight of neuroscience, but that also, as you show now, in clinical application, as you can all promise. And in some sense, it is a scientific field per se, in the sense that there's still so much to discover, right?
It is.
And we're really just at the beginning of understanding this whole system. So you're now pursuing this for quite a while. So if others would like to follow you, understanding the brain from that perspective, what is Elena's law that we should adhere to?
Just consider astrocytes and neurons and microglia and oligodendrocytes and vascular cells at the same time. So my point is absolutely. So getting astrocytes with neurons, absolutely. So stop studying cells in isolated systems.
Be inclusive.
Always try to be as complex as possible. So we need to have simple models, because otherwise we cannot really study them. But they need to be complicated enough in order to be meaningful for anything. So that's the point. So with astrocytes, we need to have great astrocytes and study circuits. We have to find the minimum circuit that is relevant for astrocyte functions, and then analyze how they interact with neurons. So that's the key idea is stop studying. You're interested in astrocytes, stop studying astrocytes only.
Right.
Collaborate with neuronal people and get outside and study astrocytes in addition to other cell types.
So stop the xenophobia.
Oh, absolutely.
Inclusive.
Yeah, that's to me. Otherwise, we won't cure diseases. Absolutely, unless we have an integral view of the brain that is based upon circuits. For me, that's very important idea.
Well, that's an interesting point, right? That you make here. You see a lot of also big pharmaceutical companies have actually withdrawn from the brain because everything they threw in.
The failure has been.
You would say that's because of this neurocentric perspective.
It's neurocentric perspective. It's not giving the drug in the right moment, depending on the disease. In the case of Alzheimer's disease, it's just all the treatments have been very late when the brain is not destroying us, it's not doing that much. But I think we need to understand I think brain are circuits, and we need to understand that and target and restore circuit function. To me, this is the key. It's not just restoring one neuron function or one astrocyte, restoring the function circuits. And that's going to be more effective on the recovery of a given disease or in the prevention of a given disease, because these circuits are impaired very early in disease. They're actually an excellent diagnostic marker for early alterations. And to me, that's the focus, the therapeutic focus and the focus for the basic research from the astrocyte point of view, from the neuron, and from microglia point of view. And NG2 cells are fantastic. We don't really know, and they do have spikes, actually. So this classification of neurons and the spike and cells that don't spike, NG2 have spikes because they are in the middle, and they're fantastic too. So
and you mentioned now at the end of our discussion,
right? But because I'm interested, so the
are they as voluminous as astrocytes?
How
are they as voluminous as astrocytes?
Yeah, they're very big. They're too very big, and they share some developmental origin.
And they talk to the astrocytes as well?
That's not known.
Okay.
It's not known. But I think we have to have comprehensive...
But why are they then differentiated from neurons if they also spike? Why were they...
Because they're not as sophisticated as neurons. They don't have axons, for instance. They don't transmit information. They don't have long distance.
Okay, but
they don't have spikes, I think. Perhaps they do because...
Does they form synapses?
I think there's a paper talking about the NG2 neuronal synapse. I'm not sure about that. I think.
Okay, that's your next talk because.
But the point for me is that we focus on our cell type or something because we need to focus on something, but don't lose the perspective that you're looking at a small thing, and that if perhaps you're missing the connections with other elements, you're missing the whole point. That's
Right.
for me, that's my point.
No, that's a point also well taken. And it's like embrace sort of this inclusive perspective, right? It's not only about only neurons, only astrocytes. Also look at this whole variable collection of cells that form circuits that we call the brain.
No, also, moreover, I talked in the talk, like neurons are highly diverse. So even saying maybe neuron is a bad word too.
Mm-hmm.
It's perhaps I'm going too far. But neuron is a bad word because if the mouse, they discover in the somatosensory cortex 30 types of different neurons.
Mm-hmm. Right.
Just there. So imagine in the whole brain, we have thousands of different types of neurons. So neuron is a bad term too.
Mm-hmm.
And if we don't understand that they're very different in different circuits, they may share some roles.
Mm-hmm.
But then now the difference may be really very relevant in the way they interact with other cells types and the way they encode and the way they store information, that perhaps that's a problem. So let's stop saying neurons.
Okay. So 4 years from now, we'll come visit you here in Barcelona in your lab, and we're going to check whether the prediction you made today was falsified or confirmed.
What prediction?
That's the question. What's the prediction that is most critical to your research that you would like to see analyzed?
That astrocytes encode specific variables in information processing. Mm-hmm. That they encode. That's the prediction. The answer may be that they don't. The answer may be that some variables that are relevant to higher brain functions are encoded by astrocytes.
Okay, very good.
That's
Elena Galea. Thank you very much for this conversation.
Thank you very much.
The CSN podcast was produced by the Convergent Science Network of Biomimetics and Biohybrid Systems, a project funded by the European 7th Research Framework Programme. For more interviews, recorded lectures, or upcoming conferences in the field of biomimetics and biohybrid systems, go to csnnetwork.eu. And thank you for listening.